My dear students of 12th class, Assalamu alaikum to all of you. Hope that you will be fine at your places. And today in your botany, we are going to take up or discuss the structure of DNA, the most important topics among this. What we are studying, that is the molecular basis of inheritance. And stay keen, as you must be remembering that the DNA is a long polymer of polynucleotides, and its structure was finally revealed through X-ray diffraction by Watson and Crick, the most famous double helical structure of the DNA, or the model of the DNA that was proposed by the Watson and Crick. And this is the double helical structure of the DNA, and what lies in the DNA, what is DNA made up of, we will try to learn about that. So stay keen. Here we go. The chemical structure of the polynucleotide chain, DNA or RNA. Let us try to first recapitulate what we know about what we have heard about the DNA. A nucleotide has three components. Nucleotide is the monomer of which the polynucleotide or the DNA is made up of. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and a deoxyribose for DNA, and a phosphate group. And there are two types of nitrogenous bases: purines, adenine and guanine, and pyrimidines, that is cytosine and thymine in case of DNA, and uracil in place of thymine in the RNA. Cytosine is common for both DNA and RNA, and thymine is present in the DNA only, and uracil is present in RNA at the place of thymine. A nitrogenous base is linked to the pentose sugar through an N or nitrogen glycosidic linkage to form a nucleoside. Again, a nitrogenous base is linked to the pentose sugar through a nitrogen glycosidic linkage to form a nucleoside, and such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine, depending upon the type of the base that is linked to the sugar. And when a phosphate group is linked to 5'OH of a nucleoside through phosphodiester linkage, a corresponding nucleotide or deoxynucleotide, depending upon the type of sugar present, is formed, because the sugars differ in DNA and the RNA. Two nucleotides are linked through 3'5' phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. The polymer thus formed has at one end a free phosphate moiety at the 5' end and 5' end of the ribose sugar, which is referred to as the 5' end of polynucleotide chain. Similarly, at the other end of the polymer, the ribose has free 3'OH group, which is referred to as 3' end of the polynucleotide chain. The backbone in a polynucleotide chain is formed due to sugar and phosphates. The nitrogenous bases linked to sugar moiety project from the backbone. And in RNA, every nucleotide residue has an additional OH group present at 2' position in the ribose sugar. Also in RNA, the uracil is found at the place of thymine, that is 5-methyl uracil. This is another chemical name for thymine. DNA as an acidic substance present in the nucleus. DNA is acidic in nature. It is found in the nucleus of the cells, And it was first identified by, as you already know, we have talked about this in the previous lecture, by Friedrich Mischer in way back in 1869, and he named it as nuclein. However, due to technical limitation in isolation such a long polymer intact, the elucidation of the structure of the DNA remained elusive for a very long period of time, unless it was only in the 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. So, one of the hallmarks of their proposition was base pairing between the two strands of the polynucleotide chains. However, this proposition was also based on the observation of Erwin Chertoff's that for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. And the base pairing confers a very unique property to the polynucleotide chains, and they are said to be complementary to each other, and therefore if the sequence of the bases in one strand is known, the sequence in the other strand can be predicted. Also, if each strand from a DNA, let's call it as a parental strand, parental DNA, acts as a template for the synthesis of the new strand, the two double-stranded DNA thus produced would be identical to the parental DNA molecule, and this is the peculiarity about this wonderful molecule, that is the deoxyribose nucleic acid, or the DNA. And because of this, the genetic implications of the structure of the DNA became very clear. And now here we have a look upon this. This is the double helical model of the DNA as proposed by Watson and Crick, and this is what lies inside the DNA in the form of these biomolecules. What is DNA made up of? That is sugar and phosphate. You can see on the other side forming the templates, and lies within are the nitrogenous bases, that is adenine and thymine and guanine and cytosine. Cytosine and guanine, as you may see, are linked through the triple hydrogen bond, whereas adenine and thymine are linked through double hydrogen bond, and then to the corresponding sugar in case of the DNA. And this is the DNA polynucleotide phosphate. You can visualize on the left hand side of your screen. Phosphate connected to the sugar at 5' position, and then the nitrogenous base that is adenine lying at the base or below. Again the chain continues, and the second one having the second nucleotide with the thymine base, and third with the guanine, and fourth with the cytosine in this form. In this way a chain is formed, and forming ultimately the polymer of DNA, beginning with 5' phosphate at the 5' end, and ending at the OH 3' OH hydroxyl group at 3' position. Oh, thanks. Have a nice day.